Let's look into paper 52, May-June 2015. Question 1. Many fungi are decomposer organisms. Decomposer means that they can break down the dead material in order to get nutrients. They carry out extracellular digestion. They break down the food outside of their body. To do this, they secrete a number of enzymes, which will help them to break down the food chemically outside their body, which they later can absorb as nutrients. A group of students made a solution of enzyme extract from a fungus. So let's say this is the fungus. Students extract enzyme from it. The extract contains the enzyme amylase. They wanted to find out the concentrations of amylase in the extract. So this is the amylase enzyme with unknown concentrations. So how are you going to find these amylase concentrations? You can perform a series of amylase enzyme dilutions from a stock or non-solution concentrations and plot a calibration graph with which you can identify the amylase concentrations that is extracted from fungi. Students were provided with a non-amylase concentrations that serve as stock solution. The concentration is 0.5 gram per decimeter cube. They were also provided with starch agar plates, with valves into which enzyme solutions can be placed. Starch agar plates are petri dishes containing agar mixed with starch. Figure 1.1 shows how the students use the plates to find the concentrations of amylase. This is the starch agar plate where you have the agar mixed with starch and they are left to solidify at room temperature. They also produce valve in the middle here. This valve, it is where you can put in the amylase enzyme. If you do a dilution from the stock solutions of amylase here, let's say you have 5 dilutions, that means you will put each of the diluted amylase into 5 different petri starch agar plate. Once you put in the amylase enzyme, plates were incubated. Now during incubations, the amylase that is placed in the valve will start to diffuse out into the agar, and they will start these reactions. The functions of amylase is to break down starch into glucose. The area where the amylase diffuse out, the starch change into glucose. After incubations, the plates were flooded with iodine solutions. Iodine is yellowish brown in color. But if they are presence of starch, it will change to blue-black in color. After the plates were flooded in iodine solutions, you can see two colors on the plate. This is the area that is stained in blue. And this is the area that is stained in brown. So if they are yellowish brown, meaning that they are non-starch. If the ionic solutions change to blue, meaning that is the presence of starch. So you can find starch here. And here it is where you will have maltose or glucose. The student thought that the area stained brown was proportional to the amylase concentrations. If you have higher amylase concentrations, the area that stained brown will be increased, because more and more starch will be broken down into glucose or maltose. So you have big area of glucose and maltose, meaning that you have higher amylase concentrations. Identify the independent and dependent variable in these investigations. Independent variable is the variable in an experiment that is manipulated. In this case, you are manipulated, you are doing dilutions of amylase concentrations. So concentrations of amylase. Dependent variable is the variable in an experiment that is measured. In this case, is the area of stained brown. Or you can just write diameter of brown zone. Describe how the student could use the method outlined in Figure 1.1 to find out the concentrations of enzyme amylase in their extract. Your method should be detailed enough for another person to follow. In this case, you need to mention about independent variable, dependent variable, constant variable, risk assessment, and reliability. First step of the experiment, prepare the dilutions. The stock solutions provided to you is 0.5. You can prepare a dilutions 0.5, 0.4, 0.3, 0.2, and 0.1 gram per decimeter cube of amylase solutions using serial dilutions. And you also need to include a control tube. In this case, the control tube you can either use a boiled enzyme solutions or use 0.0 gram decimeter cube of amylase solutions as control. Now the functions of control tube is to prove that enzymatic activity is due to enzyme but not other factor. Control tube, in this case the 0.0 gram per decimeter cube of the amylase solutions, should not have any enzymatic activity. If your control tube contains enzymatic activity, this means that the solution was contaminated. Next step, after you prepare dilutions, you also need to prepare the starch agar plate. When you prepare the agar, you should constant the percentage of the agar. Prepare same percentage of starch agar plate. And while you're preparing, you need to mix starch agar powder together, and you need to add with distilled water and also buffer with constant pH. So you can using buffer with constant pH. Number 4, pour 20 ml or pour the same volume of agar solutions in petri dish and let to solidify. So you need to constant the volume in each petri dish. This is all your constant variable. After you have poured the agar solutions in petri dish, cover the starch agar plate to prevent contaminations. Next step, after the agar solidified, use micropipette to place. You need to place the same amount of amylase enzyme in each well of the petri dish. So use the micro pipette to place, let's say 500. You can just write same volume or a fixed volume like this, 500 microliter of amylase. In each well. So meaning that you need to put the 500 microliter of the amylase of these concentrations into the first plate, this 0.4 gram per decimeter cube of the amylase in another plate, and this is in the third plate, fourth plate, and fifth plate. You need to standard the volume of each concentrations of the enzyme. And the next step is start to incubate all the 5 plates, including the plates, all the 6 plates. So you have these control plate together. So incubate the plate at, you need to mention constant temperature. All the plate must have the same temperature and for the same time. So maybe you can just put 18 hours in incubator. or temperature controlled incubator or temperature control room. Following incubations is flood the surface of the plate with iodine solutions using a dropper for 30 seconds. Pour off the excess iodine. Use ruler to examine the diameter of brown zone. And lastly, you need to repeat all the steps here to examine the unknown fungal amylase extract. So repeat steps 8 to 10 for fungal extract testing. Number 12, plot a calibration graph of known concentrations. The bin, draw a line at y-axis. So y-axis will be the dependent variable, which is the diameter of the brown zones. Draw a line at the y-axis and read off the concentrations of fungi extract at x-axis. Don't forget to mention the risk assessment of this experiment. This is a low risk experiment. Enzyme might cause skin irritations. Wear gloves during experiment. And you can also suggest how to increase the reliability of this experiment. Repeat the experiment 3 times and calculate mean. See, there are different types of amylase enzyme. They hydrolyze starch in different ways. Two of these enzymes are beta amylase, hydrolyzes every second alpha-1,4 glycosidic bond in starch molecules. And another type of enzyme is gamma amylase, hydrolyzes all alpha-1,6 glycosidic bonds and all alpha-1,4 glycosidic bonds in starch molecules. So in this case, it means that amylase enzyme got two different types. One is beta, another one is gamma. Now they are cutting off at different glycosidic bonding. So you have learned about glucose molecules. They can join by alpha-1,4 glycosidic bonding, as you can see here. So this is the alpha-1,4 glycosidic bond. But it also can join by alpha-1,6 glycosidic bonding, as such is branch molecules. So you will have this. So this is the alpha-1,6. We got alpha-1,6 molecules. Alpha-1,6 glycosidic bond, and between these, this is the alpha-1,4 glycosidic bonding. Now the question says that beta amylase hydrolyzes every second alpha-1,4. So let's say this is the alpha-1,4. This is where the beta amylase will cut off here. Whereas gamma amylase hydrolyzes all alpha-1,6 glycosidic bonds and all alpha-1,4 glycosidic bonds. So let's say if you have this gamma amylase, it is going to cut off every of these bonding and resulting in glucose molecules. So this is the one that can break and produce more glucose. This is the one that will produce maltose. In a second investigation, the students were provided with two beakers, A and B. One contained beta amylase and the other contained gamma amylase. They used these solutions to hydrolyze 25 cm³ samples of 0.5 gram per centimeter cube of starch solutions. So in this case, the students have two beakers. In these beakers, they are put in beta amylase, whereas this beaker, then they put it gamma amylase. And they also add in, so this is beaker A and B. They also add in starch solutions, which is same volume, so you have 25 cm³ and with same concentrations. 0.5 gram per centimeter cube. Now suggest and explain how the students could identify which beaker contain beta amylase and which ones will contain gamma amylase. They are producing different results. For beaker A, it is going to produce maltose. Beaker B, it is going to produce glucose. So you can just use a glucose detector. Use a glucose biosensor or glucose dipstick or glucose test strip. Which beaker that contain high glucose level, it is containing gamma amylase. Explain how. So you need to tell about because beta amylase will produce maltose mainly, while gamma amylase will produce glucose only. Question D. Humans produce the enzyme alpha amylase in their salivary glands. There may be many copies of the genes coding for alpha amylase on chromosome 1. The concentrations of alpha amylase in the saliva is positively correlated with the number of copies of these genes. So this means that if you have increased copy of the alpha amylase genes, it will increase the concentrations of alpha amylase in the saliva. So this is called as positive correlated. In a third investigation, the students obtained saliva from 6 people, A to F. So you can see in this table, you have people A to F. So this is a saliva or the enzyme extracted from person A to F. Equal volumes of saliva were added into the wells of agar plates similar to those shown in figure 1.1. The plates were incubated for the same length of time, and the area of brown zone for each saliva was calculated. This table 1.1 shows the result of these investigations. You can see that saliva enzyme extract from person A to F here, and this column show you the area of brown zone for plate 1 until 6. So they take the saliva and extract the enzyme and place in 6 plates. So plate 1 to 6 is shown here for person A. You have 3,632 area of brown zone. And you can see the lowest is the person F. The area of brown zones is only 298 millimeter square. Now, if you have increased area of brown zones, it also equals to you have higher concentrations of amylase. Also equals to you have high gene copy number. So if you look inside here, it says that if you have higher concentrations, meaning that you have high number copies of genes of genes for amylase. Identify two results in Table 1.1 that may be anomalous. Show your answer by circling the two values. So anomalous means outlier. So you can look row by row, which is the outlier. It is shown in Extract B in plate 5, 1963. And you have this 1809 on the Extract D in plate 3. State how the student should deal with these anomalies. Now the student should always remove the anomalies from replicates before they calculating mean. Eliminate affected data. From calculations. The student decided to calculate the standard deviations of their results using formula. So you have this formula. This is the standard deviations formula. Now standard deviations, this is used to measure the dispersions of data around the mean. If you have higher standard deviations, meaning you have higher spread of data around mean. Also means less reliable the sample mean. Key to symbols. S is standard deviations, X is a result. Now this is means the area of brown zone, or also means the observations. X bar is the mean. This is sum of, and N is sample size. Use table 1.2 and the formula above to calculate standard deviations for the results for person F. So we will look into only person F here. To calculate standard deviations, you will need to calculate mean value and calculate x minus mean and square it and divide by n minus 1 and square root the results. So this is the sum of x. So for the sum of x, you can add all this value. Now all this value, the x value is just come from here. So you have 298 for plate 1 and 2, 314, 314, plate 3, 4, plate 5 you have 298, and for plate 6. So this is all the value for each plate, and if you sum all this value, it will be 1820. Then you can calculate the mean. Now mean equals to sum of all the observations divided by the number of observations. In this case, it is the sum of observations is 1820 divided by the number of observations, you have 6. So the mean value will be equals to 343. Put this down. Next one, x minus mean. So you use this 298 minus mean value 303. You will get negative 5. This is just the same, so you will get negative 5, same value. And for 314, you will take 314 minus 303. You will get 11. This is same value. Then you square the value. So this is equal to 25. This is equals to 1 to 1. Now you also need to calculate the sum of this value. So you add this, you will get a value of 342. Once you get all these results, you can calculate the S, or the standard deviations. S equals to sum of x minus mean square divided by n minus 1. So the sum of x minus mean square is 342. So it is equals to 342, n minus 1 is equals to 6 minus 1, which is 5. So standard deviations will be equals to 8.27. So your answer here is 8.27. Suggest an explanations for the result shown in table 1.1. Now table 1.1 shows that enzyme extract from person A has the highest area of brown zone, and you are required to explain and suggest the results. So what is these results means? Now if a result shows the larger brown area, means the more amylase. The higher concentrations of amylase. So it is the person A had the highest concentrations of amylase, and also means that it had the highest number of gene copies. This also means there is genetic variations in populations for the productions of amylase. Some persons have higher gene copy number, some have less. If you look into here, You can talk about person F also. Person F had the lowest concentrations of amylase and lowest number of gene copies.